Okay. So last time, if I remember correctly, we did the what is that called? We did the cloning. I don't know how far you guys get. So, but if I look at the dynamic timeline, so let's share screen number 1. If you look at the dynamic timeline, you have completed the let me open my channel. So you have completed the digest, and you did heat inactivation, right? And then you did ligation, right? Right now, you followed the procedure. Hopefully everything goes well, but we don't know whether or not your ligation works, right? So what we need to do, or we don't know whether these two processes, right? The resection digest and ligation works correctly, right? In order to validate whether the process works as intended, you have to do the that diagnostic gel electrophoresis. You want to know whether your technique works, right? We can't just believe as long as I do it, we believe it's gonna work. No, we don't do that. We have to be able to see. And the way to do that is by running your gel. So I think last time we've gone through some some intellectual exercise. Let me share this one, right? Remember this one? We did this before spring break, right? We talk about the what you expect to see, and then I give you some problem set that's gonna be in your second questionnaire. So I hope you all still remember how to do this. This week you are going to run the gel for your digest. So something like this. Right here, right? So you have A plus, A minus, K plus, K minus, and the ligation. So you cannot run all 5 at once. So you have to calculate how many wells you have, because I don't know what's the gel box you have at Reseda. So try to calculate, if your gel only have 6 wells, then you will need 1 gel per group, right? Because you have 5 sample, right? A minus, A plus, K minus, K plus, and ligation, and you have to add the DNA ladder, right? I don't remember if we have ladder over there or not. Yeah, last time you run the ladder, right? Do you guys remember you have the ladder at Reseda or not? No? Can anybody tell me? No? We could. Yeah, I can't recall whether we actually have the ladder there. If not, I can bring it tomorrow. Yeah. Anyway, so yeah, so you just need to sort of like estimate the number of gel that you need, right? Depending on how much well on each gel, right? Because each group will need 5, and then you need the ladder. Don't forget the ladder, DNA ladder as well. Okay, so the idea is where is that? This this one. Yeah, the idea is on Wednesday you're gonna make your gel. Hopefully we finish with that, but and then and prepare your sample so that on Friday, the moment you come, you can just load your sample right away. Okay, so I think that's hopefully that's the plan. Yeah. Anyway, so any question about anything about what you need to do this week? No. Oh, one more thing I forgot. I think Dr. Lally will also tell you about this. There is the uh, open house, Terasaki Institute right here. So let me give you. Oh, let's see. Yeah. So let me give you the link to this here. Okay. So there's the Terasaki Open House. Terasaki is the Biomedical Institute, which is just a few blocks away from Pierce. They have the open house this Saturday. What's it called? The April 26th right here. It's gonna be from 10 o'clock until 2 p.m. So we'll be there. So the entire biotech class in the weekend class will walk actually from Pierce to the Terasaki because it's not that far. If you want to join us on Saturday, you are more than welcome to do so. I think if anything, it's probably best to park your car at Pierce and then walk there instead of, yeah, actually, I don't know whether, but if you park over there, I don't know whether parking will be an issue because, there will be a lot of people there. They're expecting about 300 plus people, 300 plus. So just if you want, again, come over here, either directly to the Terasaki Institute, or you can walk with us. Okay. So, but if you want to come, you need to RSVP. So there's a link, sign up to attend right here. You need to click this link and then just sign up. The sign up is free, but they want to know how many people are expected to show up because they have to order the food, things like that. So okay, anyway, so that's another thing for this Saturday. Okay, well, looking ahead, what I want to do today is to look ahead what needs to be done for next week, or either this week and next week. So I don't know how, and so I'm going to just give you a brief overview, remind you again the big picture, and then what's left to be done. And then hopefully, next week we can talk about the detail on transformation, and then going into, toward the end of the semester. We actually don't have much time left. After this week, you have 1, 2, 3, 4. Yeah, only 4 more, and then 4 more weeks, and then you have your questionnaire. That's it. It's very, very short. Time has passed. Okay. Anyway, so let's talk a little bit about the plan. Okay, the big picture first, and then we're gonna see what's left to do after this. Okay, so if you remember, the big picture, the main goal for this part of the activity is to produce a protein, right? Product. So let's see this one. So the product at the end. Just like any company, any business, you have to produce product or service, right? And that's how we can generate revenue, right? So the product that it can be several things, right? In biotech, it can be protein. Most of the products are protein, which a lot of your drugs are antibody, or small what's it called, a small peptide or something like that. Those are all quote unquote protein product. But you can also have small molecule, kind of like aspirin, acetaminophen, things like that, which is mostly chemicals that can be also a product of biotech or any other molecule, right? But at the end, you have to generate the product. The product that we try to make, at least in this class for us here in the biotech 2, is the red fluorescent protein. So your product is red fluorescent protein, right? In order to do so, to get to the product, first what you need to do in this series is to perform molecular cloning, right? Just to give you another perspective, it starts with the 2 plasmid, right? You have your P10R that has your RFP here, and then you have your PRA that has like what is RC over there, and you want to uh, cut your pecan R with the restriction endonuclease, which you already learned what that is. You cut it, and then you somehow insert that into PRA. So what comes up from here, hopefully, at the end, you end up with this plasmid called PRR, which already have your RFP, and it has the RRC somewhere here, RRC gene. So now this plasmid will allow your cell to produce the protein, right? So did we talk about operon at all, guys, in here? Did we talk about operon in here yet? Not yet. No. I don't see anyone say anything in the chat. No. We have not learned about operon. Okay, not yet. Okay. Yeah, we will discuss that a little bit in a moment, right? But essentially, what we need to do now that hopefully you created this plasmid after the ligation, right? After you cut them, you hit inactivate the restriction enzyme, and then you combine the two plasmid together, and then you do ligation. Hopefully at the end, you get the RR. Of course, this week you're gonna check with the gel to see if you actually have the product, right? But at the end, let's say you have the product with the PRR. What you're gonna do is you're gonna transform that into cells, okay? You're gonna put this plasmid into cell through the process called transformation. Transformation. In the transformation, now you have your E. coli cell. This is your E. coli cell. Maybe I should know. You know what? Here. You have your E. coli cell right here, and then you put your PRR inside, PRR, and then that will force the E. coli to produce your protein when the condition is right. Right? You have the molecular switch, which is what we're going to talk about, operon, right? Molecular switch that will turn on or off the expression of your RFP, red fluorescent protein, in the cell, right? So once you do transformation, the next step to do is you are going to express this protein. And that the entire process of protein production and purification is the entire thing. The entire thing is what we call biomanufacturing. Okay. So once you have your genes in the cell and you want to scale it up in the production phase, that is what we call biomanufacturing. Now, in general, biomanufacturing can be divided into two processes, right? The first one is what we call upstream processing. And of course, you can guess the second one is what we call downstream processing. What's the difference between the two? Well, in the upstream processing, it basically involves you growing your cell in a flask, something like this. Let me see. I'm sharing the right screen, right? Yeah, screen number 4. Okay. So you grow a cell in a flask, or if you are in a biomanufacturing facility, you probably do it in the bioreactor, like fermenter tank, the big fermenter tank. You sort of shake the cell, give them air, oxygen, right? So that the cell is happy, and then you give inducer. The inducer that we're gonna add. So you add the inducer at the end. Our inducer for this particular system is arabinose. The arabinose will switch the plasmid, will turn it on so that your cell can start producing RFP. And then the next day, or however long that is, after some period of time, your cell will become red, and you have to harvest them. Yeah, you basically spin them down using centrifuge. You remove the supernatant, the liquid, and then you keep that pellet. Now, I already mentioned before, in this class we use red fluorescent protein because you can see it. So when you spin the cell down, you can see your cell is red, which is kind of a good visual for us because we are still learning about this. But in the company, people don't express RFP. They express some drugs like antibody or something like that, nanobody, right? And most of them are colorless, so you can't see it. So in the real company, it's not as exciting per se. Like, you say, oh, there's this red fluorescent protein, it's glowing red, right? In the real world, it's not necessarily pretty like this, like having that red color, but it's worth a lot more, right? The antibody that you produce worth like millions of dollars sometimes, right? So, okay, anyway, so that is this whole process on producing that protein using cells is what we call the upstream processing. Okay. So the upstream processing ends after you harvest. So once you harvest, that ends right there. Okay. Now, so what about the downstream processing then? The downstream processing involves the process of separating your protein product, is protein separation, separating your product from the rest of the junk in the E. coli cell or in the cell, right? We use E. coli, we use the bacteria. So your E. coli have other protein more than just your RFP. It has their DNA polymerase, it has the enzymes involved in the glycolysis, right? It has all those enzymes, and in the downstream processing, the mission is to separate your product from all this other junk so that at the end you have a pure product by itself, not and you already remove all this other junk. Okay, so that's the process of the downstream processing. Okay, it involves a lot of technique, some biochemistry technique over there. There are different way to separate your product. So one is you can do by fractionation by centrifugation, for example. So if you're talking about company like Grifols, for example, let me just write the name of the company, Grifols or Takeda, right? You go there, they receive blood donation, so you can actually donate your blood over there to the collection center, right? They take your blood, right, and then they will separate your blood if into like red blood cell using centrifugation. I don't know if some of you may have taken anatomy and you probably learn about this. If you centrifuge your blood, right, you get majority of that would be your red blood cell at the very bottom, and then you get that white blood cell, very tiny white blood cell in between, and then you get your plasma, right? So what they did is actually they collect this plasma, right, and that within the plasma alone you have different kind of protein, right, different factor 8 or something like that. You have your albumin or something like that. So they do a lot of centrifugation method and column purification. This is like column chromatography to purify the protein. So now there are several ways of separating your protein from the rest of the E. coli protein using column chromatography. There are different techniques, right? You can separate them based on the size. That's one. You can also separate them based on their charges, like the ion exchange or something like that, right? So there are different ways to do the column chromatography, column purification. Okay, yeah. If you move on to college level, particularly once you're in the junior level in college, maybe majoring in biochemistry, you will learn like various protein purification method in a great detail, like the principle, the scientific principle behind that. There's this thing called ion exchange chromatography, affinity. There are different kind of affinity chromatography, size exclusion chromatography or gel filtration. You're gonna learn the principle behind each of these technique. For our class, it's not you're not gonna need to memorize that for Biotech 2. You will be exposed a little bit in Biotech 3. If you decide to take Biotech 3 with us, you will learn some of this through video, or you can also choose a biochemistry project, protein purification project. We actually have two students from, I think, two high school students doing biochemistry project this semester. So that will get you a heads up on biochemistry. Anyway, so those are the entire biomanufacturing process. Now, our baccalaureate degree program will be dedicated solely on this biomanufacturing process. There's a lot of things involved. There's a science portion of it, like how you operate bioreactor, for example, right? What sort of variable you have to monitor to make sure your cells are happy. In this class, you get the first introduction to biomanufacturing. We will monitor the optical density, but if you're actually in the company, not only yes, optical density information is important, but also equally important is how happy is your cell. Well, the question is, how do you know that, right? You can't just ask them with microphone, hey, are you happy? You can't do that, right? But there are some some variable that you need to track to give you a sense how happy your cells are, right? If you get too much waste, then your cell may not be too happy with that, right? So there are a lot of these parameters you need to monitor when you actually running this or doing biomanufacturing. So that's why I think half of the upper division class for our biomanufacturing program are mostly on that bioprocessing. Or maybe I should write it down. It's called bioprocessing. Basically, you grow cells from shaker flask all the way to the big bioreactor, and looking at the design of experiment, and other stuff like that. Okay, and there's also some seminar also. So that half of the upper division class in our baccalaureate degree program will be dedicated on bioprocessing. The other half will be mostly on regulatory. So we're gonna teach you about the business side of things, the regulatory quality, also supply chain, where you get all this stuff, right? So the other half of the upper division class will be on the regulatory and the business aspect of biomanufacturing. Okay, so anyway, so that's sort of like what it is. It's biomanufacturing is essentially where the money is. Well, because you start producing product, right? Remember this one? I say that at the end you need to be able to generate product or service. So that's the main goal, because that's what generates revenue. Okay, so for us, we are now over here. So you have your plasmid right here. So now we will be in the process of putting it in the cell, which is gonna be next week. We're gonna discuss the detail on the detail of this process called transformation next Tuesday, right? And then once you're done with that, you're gonna move on to grow your cells and then have them express the red fluorescent protein. Okay, and this is a good segue to talk about operon. Okay, so that's our next topic, which is the operon. Yeah, let me just write down operon. Now, just to remind you, if you look at your Canvas page, so I already opened up all the lecture activity, or the activity 7, which is molecular cloning, already there. But So the only thing you do not have is the biomanufacturing part. So that has not been published right there. So but everything else, all the way to transformation, you can see over here. There's the SOP from the Engine Biotech experience, but also you have the lecture video right here. So they're all, I don't know if you know, they're all to the 25th, which is what, this Friday or something like that. Yeah, so this Friday. So this lecture video has everything on molecular cloning and advanced techniques over here. It's about 40 minutes and 7 seconds right here. There's quiz over there that you need to do. And then there's the horizontal gene transfer over here, also 20 minutes, the 3 questions over there, and bacterial gene regulation. So in the horizontal gene transfer, basically I talk about different way bacteria can transfer their gene, including transformation. Transformation is just one of the 3 ways by which bacteria can transfer their gene horizontally, meaning between one cell to the next, right? Not one cell to another unrelated cell. So it's not from their parents, you know. So that's this one right here. And then under the bacterial gene regulation is where we are going to what is that called? You're gonna learn about the operon. And so just a quick start. What is an operon, right? So operon is essentially, it's like a molecular switch where you have multiple genes under the control of one regulatory element, right? So what does that mean, right? Well, in order to understand that, I think Dr. Lely also went through this as well. You all have learned what we call central dogma, which is the flow of information, genetic information that is, always go from DNA to RNA to protein, right? But you already learned this in biology class or something like that. So the process of making RNA from DNA is called, does anybody know? What do we call the process of producing RNA from DNA?
Transcription?
That's right, it's called transcription right here. Transcription. Okay, someone else. What is the process of producing protein from RNA? Anybody else? Translation, someone just say, okay, translation. Yes, it's called translation. Now, the reason why it is called translation is because of this, okay? If you think about DNA and RNA, they are still under what we call nucleotide. They're a type of nucleotide molecule or nucleic acid molecule, right? The monomer of nucleic acids are nucleotide. So, and there are 4 letters, right? The ATGC or AUGC, depending on whether you're bacteria, whether you're DNA or RNA, right? So this both DNA and RNA is kind of like they're the same language, if you will, which is the nucleic acid language, which consists of 4 different letters, right? ATGC or AUGC. Whereas the protein, it's a completely different language, right? The monomer is amino acid, and you have 20 different amino acid. So you're essentially converting nucleotide language, which only have 4 letters, into amino acid language, which has 20 letters, right? And that process, that therefore is called translation, right? Now, in the bacteria, you can imagine that if your bacteria, your cells are very small. You only have a limited space to contain all of your genetic material. And this is why it is in the best interest of bacteria to be more efficient, if you will, right? So if you have many genes that work or function in the same process, you may want to have all of them regulated under one control, right? And therefore, you have this so-called operon, right? So how does operon look like? Well, the general structure of operon is like this. So I'm drawing a cartoon diagram right here. Let's pretend this red line is your stretch of, RNA, something like that. Or yeah, so, or sorry, a stretch of DNA, right? So generally, the transcription process takes place in the segment of DNA that we call. Does anybody know where does transcription start? Anybody know this transcription here? Where does it start? This is for your AP test, no? Where does transcription start? Okay, someone says at the start codon. That's not necessary. That's not for transcription. That start codon is for the translation. Translation is, yes, the translation start at the start codon, which is AUG. What about transcription? Someone else says at the promoter. That is correct. So yes, the transcription in eukaryote happen inside the nucleus, but bacteria don't have nucleus. Remember, E. coli has no nucleus. So therefore, it happened in cytoplasm. But the segment of DNA where transcription start is what we call the promoter. Okay, so the promoter promoter is usually drawn as this kind of arrow, this bent arrow like that, and the arrow tell you the direction of the transcription. In this case, it will transcribe to the right. Okay. So so that's the promoter. So that's generally the promoter symbol. Now, for any operon, this behind this promoter, usually you have multiple genes. Here I'm just drawing 3, but some can be as far as like 5 different genes or so. Now, in the lecture video that that you need to watch by April 25th, that bacterial gene regulation, I talk about 2 different operon. The first one that I talk about is with the lactose operon, right? Make sure you know how that operon works, because if you decided to take Biotech 3 with us, there will be the very first activity you do is to express Taq polymerase under or using this lac operon. So the system that we use is using the lactose operon, which means the molecule that switch the gene on and off is lactose or some analog of lactose, molecule that has similar function or can be recognized with by the protein in similar way. Okay, so for that we use lactose or what we call the analog of lactose, we call IPTG. So in Biotech 3, the first thing you do is to express this Taq polymerase using the inducer IPTG, which is lactose analog. So I talk about the lactose operon in that video. We also talk about the second operon that we call arabinose operon. Now, the arabinose operon is what is the one that's relevant to our project with red fluorescent protein. Okay, so the arabinose operon, now, as you can see, I don't know if you noticed this, a lot of these operon tend to have a name of like some biomolecule like lactose or arabinose. Both of them are actually sugar, right? Anything with the OSE like glucose, lactose, fructose, anything with the OSE generally is a type of sugar. Lactose is a type of sugar. Arabinose also a type of sugar, right? There are other operon that's specific for amino acid like arginine operon, tryptophan operon. If you take bacterial genetic class upper division, you will learn about this tryptophan operon, very famous, right? But in this class, we don't worry about that. But I just want to point out that a lot of this operon is associated with some sort of biomolecule, right? Which is for a good reason, as I mentioned to you. in order for bacteria to process certain biomolecule or obtain and utilize this biomolecule, you need to have several enzymes that works together. For example, like if you in the case for lactose operon, right, the cell need to be able to take in the lactose. So they usually have these three major genes that involve in bringing the lactose into the cell and process it, right? So there are this they call it LAC, LAC-Z, LAC-Y, and LAC-A something. I don't remember what's the other one. So there are three genes that involved in helping the cell to bring lactose in and digest them, right? One of them produce like sort of like transporter. Another one is enzyme that sort of like convert lactose to be in a different form that the cell can further metabolize, right? And the other one is what you know as lactase, which is the enzyme that actually cut lactose into glucose and galactose, right? And these three genes are controlled under one lactose promoter, right? So that's why it's in the best interest of the bacterial cell to organize all the genes that works on the same metabolic pathway, if you will, under one control, one regulatory element control, right? So like all the three genes work on, bringing lactose in and digest them. It's under the control of lactose operon, right? Similarly, in the arabinose, you have multiple genes that help the cell to digest arabinose, and those genes, there are the three genes for this for arabinose. I'm just gonna draw this for arabinose. It's called ARA-B, ARA-A, and ARA-D. Okay, so and because there are three genes, ARA-B, ARA-A, ARA-D, this promoter itself is often known as the PBAT promoter. It doesn't mean it's bad or good. It's just the gene behind it is the ARA-B, ARA-A, ARA-D. Okay. Now you may be asking me now, wait, doctor, okay, how come there's B, A, and D? Where is the C, right? But the C is somewhere else. So there's this other one here called ARA-C. Right there, so ARA-C is right there somewhere else, and the ARA-C is the regulatory element. So if you watch the video on bacterial gene regulation, I even have the I also play the interactive from Lab Exchange. Does anyone know Lab Exchange? Yes. If you know about Lab Exchange, give me thumbs up or just say something in the chat. If you don't know, also say something that, okay, who does not, why don't you type it in the chat? If you have used Lab Exchange, type Y, which is yes. If you have not used Lab Exchange, type N. Let's see. Who have used Lab Exchange and who have not? Oh. Oh, all of you have not. Okay. Okay. Well, if you have not used it, it's fine. So it's actually good now you're hearing this for the very first time. Let's just look at it. It's actually really, really good resource. And I'm glad that now you're learning this right now, because it's really good resources for you to study molecular biology. So you can Google this Lab Exchange like this. You go there. You can sign in. You can make account for sign up if you want, or you can log in right here. So if this Lab Exchange was initiated by the Harvard University, right here, and they get funding from the Amgen Foundation. So the initial one, actually, it's not here. Yeah, our sponsor, I say, Amgen Foundation, right? So essentially, the Harvard University at that time have that vision of creating this open resource education, and they get the support from the Amgen Foundation. And the very first resources they have is the APE program. So all the curriculum of the APE program were, I think, the first one to be in here, and I happened to be the test group at the time. So I was one of the early educator that's in the test group to sort of like, okay, this work, this one doesn't work, and so on and so forth. But this one has a lot of resources. Now they already gather a lot more resources that even include career exploration or something like that. There's some chemistry, there's some physics now. So you see Hardy Weinberg in biology, right? Energy changers, things like that. So there are a lot more resources now than before that when they started. So this is really, really good resources. So what you can do, you can look at this. Oh, Earth and space science. Wow, they didn't have this too before, but now they have it. Good. So you can check out the library. So yeah, let's say you want to look at Opera, right? Just type over there and then some. So you see, oh, they even have trip Opera. Wow. Wow, that is good. Okay. So now they work with Khan Academy as well. So a lot of Khan Academy resources are over here as well. So here's there's lac operon right here, and there's a trip operon right here. Okay. So just take a look at no, we're not gonna do trip operon, we can do arabinose operon. Oh, there's arabinose operon right here. Is that an operon of 3 genes? Let's see. Is it the one? Yeah, I see this. Oh, this is lactose. Where's the arabinose operon? Here, the ara operon right here. The arabinose operon right here. So you can just click over here to start interactive, right? So here what I just said, right? So you have this complex regulatory element in my drawing. Am I sharing the right screen? Yes. Okay. So if, my drawing is very simplified. So look at this. It's just PBAT, and then you have 3 genes, and then there's RRC somewhere in the back, right? The reality is actually more complex than that. You have these 3 genes, RRB, RRA, RRD. Put this one away. RRB, RRA, RRD over here. This is your PBAT promoter over here. Now there is this other element that I did not discuss, right? And the RRC is somewhere in the outside all of this. The RRC is the regulatory element, right? If you remember in your plasmid, so here let me bring it back here, right? Your PRA, your PCAN-R originally, they have RFP. That's why there's an R over here, PCAN-R. But they don't have RRC there. Whereas PRA over here, that you have RRC, and this is why you want to move your RFP into PRA, because the PRA has the RRC, which is the regulatory element, right? So this RRC here is located outside. So in the case of your PRA, that's again, it's already there. Now you just put this other stuff here in the PRA, right? The entire here, the PBAT and this gene. The only difference is for our activity, we don't use RRB, RRA, RRD. We don't use these 3 genes. Instead, we take them out and we replace them with 1 gene, which is red fluorescent protein. Right? So instead of RRB, RRA, RRD, we only have 1 gene called RFP. That's behind the PBAT promoter. Okay. So what we do here is this. Okay. So the idea of the operon is you want to establish a switch, molecular switch, so that when a condition is met, your product behind that promoter will be expressed, will be turned on, right? The gene expression will be turned on. So what is that condition? That condition will be the presence or absence of that, whatever that molecule is. For us, the molecule is arabinose. So the way to switch on or off your promoter or this operon is by adding arabinose. Right? If you add arabinose, the promoter will be activated, and therefore your gene will be expressed. Right? But when you do not have arabinose, the gene will be turned off. Now, in the video, which again, I have a video there that you can watch, but here I'm just going to go through it quickly in terms of how does arabinose make this happen, make the switch on and off happen? Well, here's the story. And this is why it's good if you can have a lab exchange. I strongly recommend you to register and log in. So, you know, you can access to all these cool resources here, okay, by Harvard University. So I'm just gonna go through it quickly. You can read about this yourself. So, blah, blah, blah. So you have this RRC. So RRC is a dimer like this. Dimer means you have two protein at this, you know, bind to each other. So the RRC produce a protein called RRC also. Sorry, the RRC gene produce protein called RRC as well, right? This RRC is a dimer, and normally it sit on this so-called I1 and I2 region, right? When you have arabinos, right? So when you have arabinos, it sit there. But let's say you remove the arabinos. If you remove the arabinos from the cell, there's no arabinos. What happened here is that RRC will switch what we call the conformation. It switched the shape, if you will, right? It switched the shape to the point that it no longer bind to I2. Instead, it binds to O. Right, so it's now bind to I1 and O2 region over here, right? And by doing so, it sort of like make the entire DNA curled up like that, right? And when it curled up, it provide barrier, it provide hindrance for RNA polymerase to come, right? Because in order for transcription to happen, your RNA polymerase has to come and bind to this promoter called PBAT and transcribe the gene, right? But now that in the absence of arabinos, your RRC switched the shape and now it binds to this I1 and O2, forming this loop, right? Curling up like this. Now the RNA polymerase cannot bind, okay? And therefore, the operon is off. And this is why when you do not have arabinos, there's no expression of this gene. Remember, our gene is not RAB, RA, RAD, but our gene is red fluorescent protein. And therefore, when you don't add arabinos, you don't have red color. There's no red fluorescent protein, right? Now, what happened when you introduce arabinos? When you have arabinos, it binds to this RRC, and because it binds to RRC, it switched the conformation, or it's sort of switch the shape. Now it will release the O2, like I told you earlier, and it bind to I2 instead. By doing so, number one, it released the loop, right? So now it opens up the loop. Now it will allow the RNA polymerase to come by and start, sitting or have access to this promoter. However, a lot of promoter, they don't bind very tightly to RNA polymerase, and this is also one of such promoter. By itself, the PBAT does not bind to RNA polymerase tight enough or long enough until the transcription process starts, right? So usually the RNA polymerase can bind there, but it will come off, right? Now, however, if you have the RRC that binds to arabinos, it also help recruit the RNA polymerase. Now, there's this additional thing, the CAMP and CAP protein. Don't worry about it. You can certainly read what that is, but for our class, don't worry about it. That's another thing that will help to recruit the RNA polymerase and hold it long enough, stabilizing it in the promoter long enough so that it's there. And then finally, the the RNA polymerase can transcribe the mRNA. This is the mRNA coming up from there, okay? So again, this RRC that binds to the arabinos serve two purposes in a way, right? The binding of arabinos make two things happen. Number one, it makes the RRC release the loop, so now the RNA polymerase can get closer there. Number two, it also helps to recruit RNA polymerase there. Therefore, this is why when you add arabinos, the operon is turned on. The gene expression happen, and therefore you get your red fluorescent protein. Remember, if we don't have RFB, RA, RAD, we replace these three genes with RFP. So the moment you add arabinose, you get the production of red fluorescent protein. So I think that's the main takeaway of how this arabinose or any sugar or amino acid can can help you to regulate the expression of your protein. There are different kind of system that that we have, and this is just one of the example of that. Okay, so again, if I were you, I think now it's 35:05. If I were you, I will start looking into registering for lab exchange. This would be a good resource for you as you go to your academic journey learning upper division molecular biology or biochemistry class. They even have the career path over here, so you can also search for that. Okay. So I think that would be all. I am going to stop the recording